Welcome back to Microbial Genetics. We are now already on part 4 and on the last process of central dogma of molecular biology, which is translation. Okay, so what is translation in the first place? In simplest term, it is actually a protein synthesis. Okay, translation refers to protein synthesis. So paano? How do we manufacture protein in translation? Okay, so first, where is the location of protein synthesis? It's in the ribosomes. Okay? So in translation, we make use of RNA, mRNA specifically, no, as a template in order to create or manufacture a protein. Okay? So synthesis of proteins in ribosomes using messenger RNA as a template and tRNA as an adapter molecule that carries the amino acids to the growing polypeptide chain. Alright, so paano ba yun? Okay, so again, let's take a look at this diagram. So this is the DNA. Again, the DNA, which serves as a template in transcription, will produce mRNA. Okay, RNA will be used as a template for translation, producing now a protein. Okay? So, yun yung gagawin natin ngayon. We're going to try to make a protein. Okay? So, let's check this first, no? So, again, so this is a DNA molecule because this is double-stranded. Okay? So, double-stranded, no? This DNA molecule will undergo transcription. This is now the messenger RNA. Okay? The mature mRNA. This mature mRNA will undergo translation producing this protein. Okay? How do we know that this is a protein? Because these are amino acids, and amino acids are the building blocks of protein. Okay? This is methionine, alanine, proline, tyrosine. Okay? So these are amino acids. Okay? So bind together, they will form a protein. Okay? Alright. Ano naman tong genetic code? So, the genetic code is a reference, okay? Is a reference, no, for protein synthesis, okay? Meron tayong tinatawag na codon, no? Codon in an mRNA molecule, okay? Yung codon are three bases is consist of three bases in mRNA. For example, no, we have an later, no, you'll see kung ano yung sinasabing codon, no? Basta codon, tandaan nyo codon is consist of three bases, nitrogenous bases, no? It could be UUC, it could be UCA, UAG, basta tigtatatlo siya. And there are a total of 64 codons, okay, in the genetic code, right? So 61 codons code for amino acids, 3 codons, no, code for stop codons. Okay, so 3 stop codons, kaya 64 siya. Okay? Start or initiating codon for is the AUG. Okay, so ganito yan. Okay. This is an mRNA molecule. Okay? So how do we know that this is an mRNA molecule? By just looking at the sequence of the RNA bases because there is a presence of uracil. So we know that this is mRNA. Right? So sabi natin, codons are composed of three bases. So, AUG is one codon, AAA is one codon, GUA is one codon, CCU is one codon, GAA is one codon, ACU another one, and UGA is another one. So, how many codons do we have here for this mRNA molecule? So, we have, wait, so we have here 1, 2, 3, 4, 5, 6. So we have a total of 7 codons here. Okay? So, sasabi natin, this codon, no, will correspond to an amino acid here, no, amino acid here in the genetic code. All right? So, a codon, for example, AUG, will code for an amino acid here in the genetic code. So, paano natin titignan? For example, dito, AUG. Okay, paano natin hahanapin si AUG? So, A is the first nucleotide, U is the second nucleotide, G is the third nucleotide. Okay? So, AUG, ang first nucleotide niya is A, di ba? So, it's here. Alright? So, ito yung first nucleotide, si A. Alright? Then, we'll look for second nucleotide, which is U, uracil. Okay? So, nandito si U. Alright? Nandyan si U. Dito siya. So, dito natin hahanapin yung, dito natin isasakto yung second. So, EU. Okay? And the third codon here is G. Okay? So, this is G. So, we now have AUG. So, saan siya tumapat ngayon? Saan siya nag-intercross? Okay, so G, so ibig sabihin, AUG stands for methionine. Okay? So, A, U, and G. So, saan sila nag-intersect na tatlo? So, A, dito sa column na to. Ano? Yung A, so dito tayo maghahanap, but it's U, so dito sa column na to. And specifically, the third nucleotide is G. Okay? So, ibig sabihin is, AUG stands for methionine. Okay? And AUG serves as a start codon. Okay? Lahat ng protein starts in methionine because methionine is a start codon. No? Is coded by a start codon AUG. Okay? Nag-iisa lang. Isa lang yung start codon natin. Okay? Which is AUG. No? So, lahat ng protein, ang umpisa niya, laging methionine. Okay? Because it is coded by start codon. Okay? So, tandaan nyo, no? AUG is a start codon. Alright? Okay. So, take a look at the other example here. No? See, triple U codes for phenylalanine. Let's see if it really codes for phenylalanine. Okay, what is our first nucleotide? It's U, right? So, our first nucleotide is U. So, punta tayo dito sa first nucleotide column. So, we have U here. Our second nucleotide is U as well. No? So, dito tayo sa second column. Alright? And our third nucleotide is another U. So, kung saan sila nag-intersect? Dito. So, UUU codes for phenylalanine. Okay? So, let's take a look the GCG. Okay? GCG. So, saan natin hahanapin? What is the first codon? It's G. It's right here. What is the second codon? It's C. So, ito yung C, no? Itong part na to. So, dito tayo sa column na to magpo-focus. Alright? What is the third codon? It's G. So, saan sila nag-i-intersect? It's here in the alanine. Okay? So, it's GCG stands for alanine. Again, we have a total of 64 codons in the genetic code. Okay? So, 64 codons yun. But the 3 of them are called stop codons. Okay? 3 of the codons are stop codons. So, hanapin nga natin dito yung mga stop codons. Okay? It's actually here. No? Termination or terminal codon, no? Actually, tatlo yan sila. Alright? So, UAA or UWA, no? Para mas matandaan ninyo, no? Stop codons are UWA, UWAG, UGA. Okay? So, ito, no? UAA, stop codon. UAG, UWAG, that is stop codon. UGA, stop codon. Okay? So, para matandaan yung stop codons, UWA, UGA, UWAG. UWA, UGA, UWAG. Okay? So, those are just stop codons. And it does not code for any amino acid. At kapag yan, halimbawa, no, for example, for this mRNA, okay, uga na yung last. Okay, uga. Anong kinocode ni uga? Tingnan natin. First nucleotide is U, second nucleotide is G, and last nucleotide is A. So, it codes for stop codon. So, hihinto yung translation or yung pagmamanufacture ng protein once nakasalubong na si uga, uwag, or uwa. Okay, because they codes for stop codon. Okay? Meron pa palang animation yan. Alright? So, sige. If we're going to find everything, no? Lahat ng ng codon na nandito, si AUG codes for methionine, AAA codes for lysine, GUA codes for valine, CCU codes for proline, GAA codes for glutamic acid, ACU codes for threonine, and UGA is a stop codon. So you may want to double check, no, if I have coded, no, if I have coded these codons correctly. No, so pwede niyong tignan itong genetic code. Alright? Okay, what are the characteristics of the genetic code? Alright? It's said to be degenerate. Okay, so multiple codons may code for the same amino acid. Okay, anong ibig sabihin yan? Madaming codons, isang amino acid lang yung kinocode nila. Good example is alanine. Valine, alanine, no? Siling nyo ha, GUU, G, this one, GUU codes for valine. GUC codes for valine as well. GUA codes for valine as well. GUG codes for valine as well. So it's the generate. Multiple codons but one amino acid. One amino acid. Okay? Unambiguous. Ano namang ibig sabihin ng unambiguous? A specific codon always codes for the same amino acid. Okay? Si unambiguous, one codon. Okay? For example, UU, triple U, only codes for phenylalanine. It will never code for serine. It will never code for tyrosine. Okay? Single specific codon always code for same amino acid. Okay? Huwag kayong malilito. Sa degenerate, you have multiple codons, same amino acid. Unambiguous, one specific codon, one amino acid. Okay? It is said to be non-overlapping. Bakit siya naging non-overlapping? Because the codons are read in a continuing sequence of nucleotide triplets until a translation stop codon is reached. Okay? So, lagi siyang tigtatatlo. No? Codons are composed of three bases. So, tagtatatlo lang siya. So, it's non-overlapping. And you read it continuously, no? Wala siyang pagitan. Alright? Universal. Genetic code is said to be universal because it has been conserved from very early stages of evolution with only slight differences in the manner in which the code translates. Okay? So, lahat po ng living organisms utilize same genetic code as we are. Okay? Ito din yung genetic code ng bacteria. Ito din yung genetic code ng Homo sapiens. Okay? So, this genetic code is universal. Alright? So, paano ba nag-uumpisa yung protein synthesis or translation? Okay? So, anong nangyayari? So, we have the first step, just like transcription, we have what we call the initiation. Anong ginagawa during initiation? No, assembly of the components of translation. Okay, what are the components of translation in the first place? We would need two ribosomal units. Alin kaya dyan ang ribosomal unit? Nakikita nyo ba itong bilog na ito? Ito? This one. Okay, yung light blue na yan. No? It's the 50S subunit of the ribosome. Okay? Ito, yung maliit na ito, no, it is the 30S subunit ribosome. So we need two of them, no? 150, 130s. Another component of translation that need to be prepared is the mRNA. So saan kaya dito yung mRNA natin? This one. No, this is an mRNA molecule from 5N to 3N. Next is amino acid tRNA containing the first codon. Ano ba tong tRNA na to? Kanina pa to binabanggit, no? tRNA is this one. Itong mukhang letter T talaga siya. That is called tRNA. So, anong dala-dala na tRNA? Si tRNA, meron siyang nakakabit sa kanya na amino acid. Okay? Diba? This is protein synthesis. So, we need amino acid here. So, may nakakabit sa kanya na amino acid. No? Dito sa opposite side, may nakakabit sa kanya ng mga codons. But this is called anticodon. Okay? So, yung codons, dito siya sa mRNA. Si anticodon, dala-dala ni tRNA. Okay? Because again, if we're going to what we call this, to complement, no, the strands or the DNA bases, kailangan complementary sila. Okay? So, kailangan kung may codon ka dito, may anticodon ka dyan sa kabila. Okay? So, anong ginagawa ni amino acyl tRNA? No, it contains the first codon. Okay? So, since this is the first codon, this is a methionine. Okay? So before initiation, amino acyl tRNA synthetases, so aces, again, this is an enzyme, okay? Enzyme used during protein synthesis. Amino acyl tRNA synthetase attach amino acids to the respective tRNAs. This process is called charging, okay? So this is the tRNA. Okay, itong dalawa na to. There is a separate process actually, no? So later I'll show you kung paano. So wag muna kayong malilito. Iniipon muna natin yung mga components ng protein synthesis. Okay, so aside from these three, we also need initiation factors such as IF1, IF2, and IF3, and of course, energy source, which is GTP. Okay? So, isa-isahin natin itong diagram. Tignan natin kung lahat ng components na kailangan nandito na. First, the two ribosomal units. Meron na, may 50S, may 30S dito. Nandito na rin yung mRNA. Amino acyl tRNA, this is tRNA and contains amino acid already. Okay, initiation factors, no? Nandyan lang yan, pakalat-kalat si initiation factor. Alright? GTP and energy, no? Kailangan ng energy during translation. Okay? So, kung mapapansin nyo dito sa 50S ribosome, meron tayong tinatawag na A site, P site, and E site. Okay? Si A site, it is the binding site for incoming amino acyl tRNA. Okay? So, si P site occupied by peptidyl tRNA, and E site is occupied by the empty tRNA. Okay, so this is the E site, P site, and the A site. Alright. So, ang process ng translation, guys, tawag dito, is still 5 to 3. Okay, 5 to 3 pa din siya. So, sabi natin kanina, no, tRNA and the attached amino acid needs to be activated first. Okay? So, this is the enzyme amino acyl tRNA synthetase. Okay? So, ito yung responsible sa pagkabit ng amino acid sa tRNA. So, this one is amino acid, no? And it makes use of ATP to activate the amino acid tRNA. Okay? Ayan. This is our tRNA, no? tRNA siya. No? So, magbabind din siya dito sa amino acid tRNA synthetase para kunin yung amino acid. So, we now have amino acyl tRNA. Amino acyl tRNA. And it is one of the components of RNA synthesis. Protein synthesis. Alright. So, di ba, we now have assembled the components needed for tRNA. Okay, so ano yung unang mangyayari? No, ano yung first step or first in the process? Recognition of ribosomal binding site. Right? Remember, during transcription, there is DNA promoter sequence, no? Sa translation, meron tayong tinatawag na ribosomal binding site where the ribosomes binds to start the translation or protein synthesis. Okay? So, ano-ano yung mga ribosomal binding site na meron tayo sa prokaryotic mRNA? We have the Shine-Dalgarno sequence. Okay? It is located upstream to the start codon AUG. Okay? So, take a look at this diagram, no? Kita nyo ba to? This is AUG, right? AUG is start codon. Sabi ng definition, the Shine-Dalgarno sequence is located upstream the AUG. Okay? So, dito po siya sa unahan ng AUG. So, this is the Shine-Dalgarno sequence, the yellow one. Okay? So, anong ginagawa ng Shine-Dalgarno? It helps recruit ribosome to the mRNA to initiate protein synthesis. Okay? So, yun yung trabaho niya. On the other hand, for eukaryotic mRNA, ano naman yung kanyang ribosomal binding site? Capped 5 end. Okay? So, this is the 5 end, no? And this end is already capped. Okay? So, ribosome advance to AUG through linear scanning. Okay? Kapag nakita ito ng ribosome, itong capped 5 end, aabante yan siya para mag-scan hanggang makita niya si AUG. Again, AUG is a start codon. Okay. So let's say the ribosome already found the ribosome binding site, no? So nakita niya na kung saan siya mag-start. Okay? And the first amino acid, tRNA, already binds to the P site. Where is the P site? This one. Yung light blue na to, yung rectangular, that is the P site, no? And dyan unang nagbabind yung tRNA. Okay? Alright. So to elongate the protein, okay? So, 'di ba, meron na tayong first tRNA here, no? At may kasama po yang yung tRNA na to, may kasamang amino acid yan diyan, 'di ba? So, if you already have amino acid there, kailangan mag-form ng peptide bond. Peptide bond is the bond linking the amino acids to make a protein. Okay? Catalyzed by peptidyl transferase enzyme. No? So, ito yung enzyme na gumagawa ng peptide bond. Okay? Once meron ng peptide bond, itong first tRNA will move here to the E site. Okay? And then, the second, okay. So, let's say nandito no, sa P site yung initial tRNA. The second tRNA with amino acid will enter through the A site. Okay? Ito siya, binding of amino acid tRNA to the A site. Okay? Remember, yung hindi lang nagbabind sa A site is the initial TR or the first amino acid tRNA kasi dito siya kaagad sa P site. Okay? But the subsequent amino acids or tRNA always um, placed here in the A site. Okay? Alright, so kapag meron na kapag nandito na yung first amino acid, second amino acid, nakapag-create na ng peptide bond, magkadugtong na yung first amino acid sa second amino acid, umuurong po ito. Okay? So, ayan. Umuurong siya. No? So, this is the first tRNA, no? So, napunta na siya dito sa E site. Alright? tRNA na lang ito ha. Nasaan na po napunta si amino acid? Nandito na sa taas, nakadugtong na siya sa second. Okay? Nakadugtong na sa second. Alright? And the incoming tRNA will again bind to the A site. Tapos, itong first, ma-expel na siya. Sa second, pupunta na sa E site. And then, si third amino acid to sa third tRNA, pupunta na sa P site. Okay? And it goes on hanggang humahaba itong nasa taas. So, ito yung babantayan ninyo. Dapat humahaba or dumadagdag yung amino acid dito sa taas. No? So, that is elongation. Alright? So, paano naman titigil yung elongation? Okay? So, nakita nyo? This is, mahaba na yung amino acid, no? Sorry. Kita nyo? Mahaba na si amino acid. Sorry Alright. So, kita nyo mahaba na si amino acid here. So, how many amino acids we already have here? 1, 2, 3, 4 amino acids na magkakadugtong. Anong bond ulit yung nagdudugtong sa amino acid? Peptide bond. Okay? Anong enzyme yung nag-create ng peptide bond? Peptidyl transferase. Okay? So, ayan o, humahaba na siya. And then, si tRNA nagmo-move lang ng places. Okay? Para ma-accommodate yung incoming tRNA. Alright? So, yung termination occurs when one of the three termination codons moves into the A site. Okay? Ano nga ulit yung tatlong stop codons natin? Uwa, uwag, uga. Okay? Once nasa A site na yung isa sa kanila, mag-initiate na ng termination. Okay? So, results in the release of the newly synthesized protein and dissociation of the ribosomes and mRNA. Again, the green one is the mRNA, di ba? Okay? And this is the amino acid. Itong blue na ito are the ribosomes. Okay? So, again, kapag na-meet ng A site, nakapunta na sa A site, yung termination codon, automatic, the synthesis of proteins stops, no? Alright? At meron din tayong release factor dito. Meron ding release factor dito, no? So, ayan, yung red na yan. Para ma-release totally si formed protein. Okay? So, this is now the free polypeptide. This is the free tRNA. Okay? So, termination, ito results in the release of the newly synthesized protein. This is the newly synthesized protein and dissociation of the ribosomes and mRNA. These are ribosomes, no? Ito, ribosome, and this is mRNA. No? If they are already dissociated, then protein synthesis is done. Right? Okay. So, let's, what we call this? Tapusin natin yung isang, ano, no? Isang central dogma. Okay? Isang gene expression from DNA molecules which will undergo transcription, you now have RNA. Okay? So, ito si RNA sequence mo. GCU, ACG, GAG, and so on. So, again, we have codons, no, which comprise of three bases from the RNA molecule. So GCU is codon 1, ACG codon 2, GAG is the codon 3. Again, these codons have corresponding amino acids. Saan ninyo makikita kung ano yung tamang amino acid dito for specific codon? You have to look for the genetic code. Okay, titignan nyo si genetic code. Alright? During translation, we need a tRNA. Anong trabaho ulit ni tRNA? Siya yung nakadikit sa kanya in one end, nakadikit sa kanya si amino acid. On the other end, si anticodon. No? Anticodon because magkabaliktaran sila ng codon itself. This is the codon, right? Sabi natin, anong laging kapartner ni guanine? It's always cytosine. Kapartner ni cytosine, always guanine. Kapartner ni uracil sa RNA, always adenine. Okay, so anticodon siya, kabaliktaran ng codon. Alright? Why is it necessary? Because as we all know, the tRNA binds to RNA molecule, no? Depending on the corresponding codon. Okay? Alright, so these are the anticodon for these RNA, for these codons, no? Ito sila. Alright, so sabi natin, si tRNA may kasama siyang amino acids on the other end of the molecule, no? So GCU, that is the codon. Ano yung corresponding amino acid niya based on genetic code is alanine. ACG correspond to threonine. GAG correspond to glutamate. CUU correspond to leucine. Okay? So, wag kayong malilito kasi baka yung hanapin ninyo sa genetic code is the anticodon. Okay? CGA does not code for alanine. Okay? So, yung lagi nyong titignan sa genetic code is the codon or the RNA molecule itself, not the anticodon. Okay? So, ganyan po gumawa ng protein. Okay? Medyo complicated yung pinagdadaanan niyang process, no? But, it's simple. Okay? Kailangan ng template. Again, no? Just to fulfill the concept. Again, everything, no? Every information about the structure or characteristic of organism are stored in DNA bases. So, DNA bases, no, contains the blueprint of an organism. Okay? So, in these DNA bases lies the gene. No? What is a gene again? Gene is a basic unit of heredity. Okay? And gene is a functional structure. No? It codes for something. It codes for protein. Alright? So, for example, these are all included in a functional gene. So, we need to transcribe this to RNA molecule. This RNA molecule, no, once transcribed, is not final, no? It needs to undergo post-transcriptional modification. Okay? Tatanggalin yung mga introns, ititira si exon. Alright? Itong exons, or what we call the mature mRNA, will be divided into codons, no? Into three bases. Because these codons code for amino acid. Okay? Anticodon is needed because si tRNA, para makadikit siya sa RNA, kailangan complementary din sa codon yung mga bases na dala-dala niya. Okay? So, if we have already get the amino acid, no, nag-undergo na ng activation si tRNA, dala-dala niya na yung amino acid, no? And then, tuloy-tuloy na yung pagdadagdag ng amino acid during translation until the ribosomes or until the A site would contain the codons, no, or the stop codon, no, doon lang matatapos yung translation process. Okay? So, hope pwede nyo namang balik-balikan yung video kung malilito kayo, no? So, at the end of this lecture, I want you all of you should be able to transcribe a DNA base into our RNA and synthesize a protein coming from the mRNA molecule. Okay? So, ano bang sense or ano bang importance ng pagdidiscuss ng microbial genetics to nursing students, no? These have clinical correlates, no? Hindi lang ito, it's not enough for us to know the technicalities of genetics, no? It has clinical correlates because we are healthcare professionals and we have to apply, no? And we need to apply these learnings, okay? So what are the applicable clinical correlates here? If you are familiar with Amanita mushroom, the poisonous mushroom, okay? So, the scientific name is Amanita phalloides or known as death cap, no? So, Amanita phalloides contains peptide toxin alpha, termed amanitin, okay? Which inhibits the RNA polymerase 2. Saan ninyo naaalala si RNA polymerase 2? Anong process? Saan ba siya present, no? It is present in transcription. Alright? Transcription. It is responsible for the synthesis of RNA. Alright? So, if you happen to ingest Amanita mushroom, okay, it will inhibit inhibit the function of RNA polymerase 2. If you have if your RNA polymerase 2 is absent, what is going to happen? Magkakaroon ka pa ba ng transcription? Wala na. So transcription will not push through as well as translation kasi wala ka namang finish mRNA product. Okay? So yun yung what we call this importance, no, of learning the specific enzymes, okay, because it has clinical application. See, pag may pasyente kayo tapos sa based on history nyo, nakakain po ako ng poisonous mushroom, you would know na na-inhibit nito yung RNA polymerase 2. So what are the clinical symptoms of Amanita mushroom poisoning? Gastrointestinal symptoms, acute liver failure, and it can be fatal. No? It can lead to death. All right. So what else? Diphtheria toxin. Who are familiar with diphtheria? Okay. So diphtheria toxin are produced by Corynebacterium diphtheriae. Okay. So these are gram-positive bacterium, no? So ang pinaka-common presentation nito is pseudomembrane formation on the soft palate, hard and soft palate, no? So, napakakapal na pharyngeal membrane yon. Alright? At tapos, meron silang bull-neck appearance na malaki yung lieg nila. Alright? Kapag meron silang Corynebacterium infection. Okay? So, ano namang ginagawa ng toxin ng Corynebacterium sa genetics, no? Or sa DNA molecules? So, ang ginagawa, it inactivates the eukaryotic elongation factor or EF2, preventing the translocation. No? Saan natin makikita si EF2? During translation process, during the elongation, where the tRNA translocate into the next site, no? Kailangan ng elongation factor 2 doon. And if it is being blocked, no, or inactivated by diphtheria toxin, translation will not go through. Okay, wala, may mapuform ba na protein? Wala. And it will affect the organism. Right? So what is the clinical manifestation of diphtheria toxin? Results in local tissue destruction and pseudomembrane formation. Alright. Another set of clinical correlates the use of antibiotic. Okay, what is antibiotic? Anti, no, biotic, no? Ibig sabihin, what is biotic? Living organism, no? So, antibiotic could kill microorganisms. Alright? So, what are the most common antibiotics in related to microbial genetics? First is rifampicin. What is rifampicin? This is a TB drug. Okay? Remember, yung TB drugs, di ba, they are four drugs, no? For maintenance intensive phase, the patient takes four types of drugs, no? So one of which is rifampicin. Okay, ano bang trabaho ni rifampicin? Ano yung mechanism of action niya? It binds to the beta subunit of bacterial DNA-dependent RNA polymerase. Saan natin ulit nakita si RNA polymerase? During transcription. Okay? Magkakaroon ba tayo ng RNA product kapag wala tayong RNA polymerase? Of course not. No? Kapag wala tayong RNA product, wala ding protein. At kapag walang protein na napo-form, walang magagamit yung bacteria to function as a food. Okay? That's why rifampicin is a very, very good drug, okay, against microorganisms, specifically Mycobacterium tuberculosis. All right? Okay. What else? Let's proceed here on the other antibiotics. Are you familiar with chloramphenicol or tetracycline? Okay. These drugs affects protein synthesis or translation. So, tignan ninyo kung ano yung sinisira ng mga drugs na ito doon sa process. For example, streptomycin and aminoglycosides binds to the 30S subunit and interferes with initiation. Okay. 30S subunit is a ribosome where protein synthesis happens, right? So, kung yung 30S mo, hindi na makapag-function, makakapag-protein synthesis ka pa ba? No. Okay? And kapag walang protein synthesis, the bacteria won't function, no? Kapag wala silang protein products na pwedeng gamitin, the bacteria won't function. Okay, that's why streptomycin and aminoglycosides is a very, very good antibiotic. Alright, tetracycline inhibits or prevents binding of amino acyl tRNA to the A site. So, saan ito? Elongation process din. Alright, or elongation step ng translation. Si chloramphenicol naman, anong trabaho niya? It inhibits peptidyl transferase. Ano ulit yung trabaho ni peptidyl transferase? It is responsible for peptide formation between amino acids. So, ito yung taga-bind ng amino acid in forming proteins. Okay, how about clindamycin and macrolides? Kung si streptomycin and aminoglycosides bind to the 30S, clindamycin naman binds to 50S subunit, inhibiting translocation or translation as a whole. Okay? So I hope you have a full understanding, no, of the basic of genetics in a microbial organism. Okay? So if you have questions, feel free to message, no, or you can send it through email.